So, if we draw a cell, most of you probably do something like this. You do something that has a line around it. My guess is no one here knows what the line is made of. You've been drawing this probably for 3 years, 4 years, right? And not really understanding what it does and what it is, just drew it. Correct? All right. So the inside is made of organelles. What's interesting is that the outside of the cell has water, because water is the universal solvent, it's a life solvent. The solvent of life is water. So if you're talking about living things, there's going to be water outside the cell, and there's going to be water inside the cell. So this line that you all drew in your morning assignment, because I saw them, you all pretty much drew this line. Some of you labeled them, some of you didn't. Some of you drew like bacterial cells, some of you drew an atom, which is not correct. Some of you drew all kinds of other things, so cell dividing. Some people drew balls, little spheres. But a lot of you drew this little line. All right, and this line, say what? What's going on? No? I get confused sometimes. So there's this line on the outside of the cell. This acts as a separator, right? As a divider, as a blocker. It creates a what? What does it create when you draw this line? And we call it the plasma membrane, by the way, or the cell membrane. Another word for this is cell membrane, or plasma membrane. So there's a cell. The plasma membrane is a separator, right? It separates what from what? When you drew that line, what did you do? People, I want you to say something. Yeah. Well, let's not call it a bacteria, because actually this is not a bacterial cell, right? This is actually a eukaryotic cell. We'll talk more about that in a little while, probably tomorrow, okay? But right now I want you to think basically, very basically. When you drew this line, just fundamentally, what did you do? What'd you do?
Like put like a wall around.
Well, let's not use the word wall, and it's a good word. It's a good word, but I don't like to use it here, because plant cells have walls. Fungal cells have walls. Bacterial cells have walls, but your cells don't have any walls. So let's not use that word. Let's use a separator, divider, or blocker. Can you think of another word that's not wall? Or one of these three? A barrier. I like that one. I couldn't think of it. I didn't hear you say barrier. I recorded this, so I got evidence that I didn't hear you. Barrier. So a barrier, a separator, a divider, a blocker. Well, what is it dividing? What is it a barrier against? What is it blocking? What's it dividing? Yeah.
What is the green pointing to?
The green is pointing to that line that I'm talking about.
So what is the
That line,
is too?
this line here, see this line right here?
And the
The white line is a separator, a divider, a blocker, a barrier.
and then the cell membrane.
It's also called a cell membrane or a plasma membrane. These are the names that this thing is called. This outer line, this outer line here of this thing that we call a cell, right? It's called a cell. Remember, what does a cell mean? What does the word cell mean? Oh, it's going to be on a quiz tomorrow. There will be a quiz tomorrow on this work, by the way. A room, that's right. It means room. So this is a room, a space, this inside space. So what is this separating? Very basically. From other cells, but if it's a single-celled organism, it's just floating in a stream, what's it separating? You're going to kick yourselves when I say this, but I'm going to go ahead and say it. No, that's a good answer. It's separating the water. Water where and water where? Inside versus outside. So you have an outside and you have an inside. So now you have water outside and water inside, but only because you have a room, right? Only because you have a separator, a divider. Does that make sense? Okay. So that cell membrane acts as that barrier from the outside. Now I'm going to use an analogy, and people, when I use analogies, people forget to use these analogies on tests. I don't say these stories to help to confuse you. I'm helping you try to understand the idea. And here's the idea. You exist right now in a room. You probably live in a house. When you drive in a car. All these things have separators, blockers, dividers, barriers, right? This room has barriers. What are those barriers? The walls in this room. And why are they important? What do they do? What do they allow us to do? They separate outside from inside. But what does it allow us to do? Protect ourselves is a good word, but there's something else. What does that protect us against? What? To do what? This inside versus outside. If it's cold outside, what can we do inside? Stay warm. If it's warm outside, what can we do inside? If it's wet outside, what can we do inside? And if it's dry outside, we can have a pool in here, right? Indoor pool. So we can be wet inside when it's dry outside. Our bathtubs, for instance, right? So the conditions can be different. So the separator, barrier, or divider. I'm not using wall. Do not use wall. It's going to confuse you later. The separator, barrier, or divider allows the inside of the cell to be different. So if it's acidic outside, does it have to be acidic in the cell? In fact, you have a stomach, don't you? And all of you ate breakfast, or most of you ate breakfast, or will eat lunch, or will eat dinner. At some point today, you'll probably eat, or you'll pass out, but you'll eat something, correct? And as soon as you eat something, what's going to happen? What does your stomach do? How? It creates extremely acidic conditions inside that cavity, inside that room that you call a stomach. Right? Because all life creates rooms, creates compartments. That's what we do. We create compartments. Why? So we can create places that have different environments, different conditions. So inside that stomach, it gets really acidic. So all that stuff that you ate, you know, when you eat a sandwich, you eat a hamburger, do you know that's made of protein, right? It was a cow. Do you know that? And that cow had skin and muscle like you, correct? And when you put that in your stomach, you break that skin and muscle down, don't you? Well, why doesn't that break you down? The barrier. The cells repel that. They produce this mucus, and we can talk about it later if you like. But the bottom line is that this membrane allows for the conditions on the outside to be extremely different from the conditions on the inside. But this is a very special, this plasma membrane is very powerful. Without it, life could not exist. So this barrier, this plasma membrane is what allows the inside of the cell to be different from the outside of the cell. And somebody said separate from other cells. That's correct, but it separates it from everything. Inside every cell, there can be a completely different set of environments. And not just this cell, but then inside every compartment can be different, right? So that's going to be key. It's going to be very important you get that idea. I can tell you, I'm going to put it on the quiz. So because these barriers, these separators, these dividers create different rooms, right, different compartments, the cell is the big room, right? So this is a cell. It's a big room. But each one of these organelles is another little room we call compartments, all right? So by creating compartments, we can create different conditions, different chemical conditions. They can be acidic, they can be basic, they can have a lot of enzymes that break things down. In fact, when you die, there are these organelles in your cells that are called lysosomes that have enzymes that break down everything. And when you die, those lysosomes bust open, those enzymes are released, and you turn into slime. And they break you down. That's why, in fact, I used to watch these scary movies. I don't really watch really super scary movies because I have nightmares. But I used to watch Buffy the Vampire Killer or something like that. I thought it was a little scary, but I thought it was cute. So anyways, Dracula and all that, and they would make jokes like, oh yeah, that's a wet one. When they would open up the dead, Bones. You ever watch Bones? It's a little gross, but I watched the whole show like 3 times. I love it. So
I
yeah.
watched like the entire thing 4 times.
Yeah, I have too. My wife and I watch it literally, well, not 4, but 3. We'll catch up to your mother. We'll watch it one more time. But the interesting thing is they would call a fresh dead body that's been sitting underground for a while, they call it the wet one. Well, it's wet because those lysosomes broke open and they broke down all that meat, all that protein, all that fat, all the DNA is all broken up into this mush. But that's only because those little compartments in the cells broke open. Because when you're alive, you keep those conditions contained in a little vesicle, a little compartment. But we'll talk more about that later. There was just an example of this idea that these separators allow you to have different conditions inside each of these spaces. Remember that a lot of you have drawn this, but you don't even know what they're made of. Can anybody guess what they're made of? So let's think about it. I bet you could guess. I bet you would know if you read the chapter, which, by the way, is in your syllabus. It tells you what chapters you should have read it last week. I know you don't read the syllabus, I understand that, but it does say it. It says it clearly in green, right in the middle of the syllabus. You can look at it on Schoology, what chapters you should have read or should be reading. But let's think about this. If you're separating outside from inside, outside's water, inside's water, and keeping those separate, what qualities does this cell membrane, what qualities must that cell membrane have? If I'm asking you a question today and I'm telling you you have a quiz tomorrow, maybe it's important. Yeah. Hydrophobic, excellent. This has to be hydrophobic. Because otherwise, water would dissolve it, right? So this thing has to be hydrophobic. But then it also has to be what? Because it's interacting with water on this edge, and on this edge reacting with water. What must it also be? Hydrophilic too. What do you know of that's both hydrophilic and hydrophobic? Phospholipids. To remind you from yesterday, phospholipids, where did I draw it? I thought I drew it. I bet you it's here somewhere, and I don't want it.
Isn't it in the corner?
Kind of. So it has to be both hydrophobic and hydrophilic. It has to be both. I'm going to find it later. I'm going to be very upset. So here it has to be hydrophobic. That's the tails. The 2 tails have to be hydrophobic. And the head has to be hydrophilic. I want to be clear on this. When I say head and tails, I want to remind you, there were 3 carbons that were part of the glyceraldehyde, or a glycerol that became a glyceraldehyde molecule. On one of the carbons, among other atoms, they're attached to this group of atoms called a phosphate. That phosphate is negative. It's charged, which means, I want to remind you again, that charged species is also known as hydrophilic. And hydrophilic means it loves water, mixes with water easily. But then it has this yellow side, let me draw it in yellow, that is a chain of carbons and hydrogens. Carbons and hydrogens, I'm just drawing this little line, but at every corner there's a carbon and two hydrogens all the way down, 3 hydrogens at the last carbon. And then this one is called a saturated fatty acid we've learned about in our last unit, and the key word here is fat. It's a fatty acid. Then it has another fatty acid. Sometimes might have a double bond. Obviously it says kind of ridiculous number of bends, but this other fatty acid is a double bond, which means it's a what, by the way? What's this one? It's a fatty acid, but what is it if it has a double bond? Did I ask you to draw that? It was on my multiple choice, but not on the essay. Yeah.
Is it saturated? I mean
Unsaturated. Fatty acid. And again, the clue here is that you're dealing with a fat. So because it's a fat, this yellow part is considered what? Not hydrophilic, but hydrophobic. It's not hydrophilic, it's hydrophobic. So I also want to remind you, we talked about this thing called micelle yesterday. And yes, there will be the yesterday's stuff will be on there as well. These micelles are balls of fat, right? They're literally balls of fat. The inside is hydrophobic, they're the fatty acid tails. The outside are the phosphate heads. This is not a cell. We call it I want to remind you, we call it a micelle, but it's not a cell. That's why I don't like the word micelle, because kids get confused, people get confused because the word cell is in there. But it is not a cell. Remember, a cell has a space inside. A micelle doesn't have a space. It doesn't have any water inside. A cell has water inside and outside.
They can't change the name?
Well, it's not going to happen, so don't worry about it. They can do whatever they want, but they're not going to do it. So phospholipid bilayer. Oh, that's the thing. This phospholipids can't form a single layer that's all smashed up into a ball. It has to create a space, has to create an envelope. Remember, it's 3-dimensional. It goes all the way around the cell. The cell is round, it's spherical. It's not round like a circle, it's spherical. So it goes in both, it has depth, it goes all the way around. I cut it open, this is cut open. This is a slice through the cell. So don't get confused about that. So when we look at this, these are not made of phospholipids, they're made of a phospholipid bilayer. A bilayer. And what the bilayer looks like is this. There's 2 layers of phospholipids. So this divider, this separator, this plasma membrane, this cell membrane, a lot of the membranes that create compartments are going to be 2 layers. 1 layer of phospholipids here, 1 layer here, allowing you the phospholipids, the tails to mix, allowing water to be on the outside and water to be on the inside of what you're creating. Does that make sense to everybody? So that's pretty much a phospholipid bilayer, except you got a problem if you just create that. What's the problem? What's the problem with this membrane just being a divider, a blocker, a barrier? What's the issue? This thing's alive, isn't it? What's the problem with that being just a blocker if it's all the way around the outside of this cell, all the way around is just a blocker, a separator, a barrier? Yeah. Yeah, you can't get any food in. You can't get any materials in, and you can't get stuff out. Look, if something's alive, and all of you know this, they got to eat and they got to poop. Why is your notebook? Are you taking notes? Okay, I can't see you. Sorry. It looked like you were at the notebook closed. I was like, what's going on? So things can't get in or out. So what do you need? What do you need? I want to remind you, I said this earlier, but I know some people came in late. Notes from now on are going to be graded strictly on Cornell notes. So if you're not putting in a summary, when I ask you to submit some stuff, sometimes they won't have summaries. I can live with that because if I ask you to submit something that, like today, if I said turn in your notes, obviously you haven't had time to put summaries. But I want to see details. I want to see actually good notes put in. All right? All right. So phospholipid bilayers, guys. Oh, so we said things have to get in and out. Do you understand that? You have to get food in, and you have to get poop out. You have to get waste out. Waste has to come out. Not just waste. Your acid in your stomach that we talked about earlier is produced by the cells of your stomach. You have to get that acid into the stomach. You all, when you smell something good, your mom's cooking something delicious in the kitchen, and you're hungry, what happens in your mouth? It waters. It's saliva. We just like to say water because it makes us feel better, but it's spit. So our mouth fills with saliva. Why does it fill with saliva? How does that happen? Where does that saliva come from? Inside the cell. Those cells produce the saliva. You have hair on your head, right? Where'd that hair come from? It grows every day. It's not alive. This is not alive. This is a protein. Where's this protein coming from? The cell. There's a cell at the base of your scalp that starts to produce the protein and stick it and keep sending it out. So all the stuff that you make has to be made inside a cell and then put out of the cell. So you have to and you have to also get rid of the waste. You pee, don't you? In that yellow stuff in your pee is the waste from the cells. So everything that's in the cell that your cell doesn't want or your other cells need have to come out of a cell, and stuff that the cell needs has to go into the cell. So we have to be able to get things in and out. So it can't just be a barrier, a divider, a separator. It can't just separate. It must also do what? What else must this cell do? It's alive, so what must it do? Yeah. Take in stuff and put stuff out. Now, is it going to just take everything in? So it has to be selective, correct? Well, letting things in and out is a word that we use that we say it's when we say in and out, we call that permeable. Permeable. That means it's something's allowed, things are allowed in or out. But we call it selectively. Selectively permeable. It allows certain things in and out. It chooses what to allow in and out of it. Like your house. You don't still let anybody walk in. Right? I hope. Only people that are allowed to walk in. So selectively permeable. Another word that some people like to say, I'm going to put it because you might read it in a book, is semi-permeable. I don't like that one so much because it suggests that it's half permeable, but It's not true. It's selectively permeable. Are we all okay with that?
Mm-hmm.
So what is that? In order for it to be selectively permeable or allows things in and out, what do you need to have? Yeah. Okay. So you got this wall, you got this not wall, you have this divider, separator, barrier. What has to be in that divider, separator, barrier for this to happen? A what? A what? Well, DNA's in there, but I'm not sure how does that help you get things in and out of this barrier, divider, blocker. Like, what do you need to put in this thing to let you get in and out?
I think she meant DNA because DNA carries information, so it goes in and out.
That's a good point. Information that's a really, wow, it's really complicated. Well, it's not really that complicated, but it's really interesting. But we're not at that point where we can have that discussion, but actually DNA stays in the nucleus in eukaryotic cells. It does send the message out, you're right, but in a very special way that you're going to memorize. So you're going to know. That's going to be one of the more important aspects of how that happens, okay? But everybody try to focus here. You got to get things in and out. This is a divider, a separator. It's partly hydrophobic, partly hydrophilic. It surrounds the whole cell. We're saying that the cell is semi-permeable or selectively permeable. It's going to allow some things in, some things not. So what do you need for that to happen? A door. You need some kind of opening, right? You need an opening. Good, good. So you need an opening. Well, what are you going to make the opening out of? Say that again?
What are you going to make it out of? I was about to write that in the
Oh, as a question. Well, you go ahead and write it. What are you going to make it out of? What are you going to make the door out of? Make a guess. The water. Okay, it's an interesting idea. But and you got a problem. If you're going to build a door, and this is the membrane, right? And this is hydrophobic here. Let me change the color. Let me zoom in as well. If this is the membrane, right? It's made of hydrophobic and hydrophilic stuff. This is hydrophobic. You're going to put a door here. If you made it of water, what's the problem? It won't stick in this area, right? So it has to be at least partly hydrophobic. Also has to be partly hydrophilic. Yeah. Proteins, correct. Proteins are very interesting little things, aren't they? Water inside and outside, but the hydrophilic head's outside and the hydrophobic tail's inside. These membranes we need proteins. Remember what a protein looks like. Protein looks like this, right? And where the amino acids look like these. Amino acids have this backbone that's the same for all of them, right? And then you have these R groups that stick out. They can be polar, they can be non-polar, they can be charged, or they can have rings, right? These straight chains, they can be small, they can be big. They can be aromatic. So anyways, these R chains can be different. The key here, though, is that a lot of them are non-polar. A lot of them are non-polar. But some of them are charged or polar. The charged or polar, another word for them, for the charged or polar is what? Charged and polar both are called what? They're both hydrophilic. So charged and polar are hydrophilic. That's what's great about this word hydrophilic. It means polar and charged. The non-polar what? Hydrophobic. So non-polar are hydrophobic, the polar are hydrophilic. So you can take these and put a lot of them in a chain, in a long chain. Remember, we're going to take these, the backbones, we're going to stick them together using peptide bonds, using dehydration synthesis and creating peptide bonds. We're going to put them together. Some of them are going to be polar. Notice they're all different shapes and sizes. But the blue ones are polar and the yellow ones are non-polar. Well, that's very important because what that allows us to do is when we get the membrane, it allows us to stick this protein in like this. Do you see that? You see how the non-polar can now stick with the non-polar, and the polar can stick with the polar, and now you got, and if you did two of these, if you had two of these, and you had two, and obviously this is continuous. I hope you understand that this, I hope you understand that this is continuous, so the membrane goes out both sides. What did you just create? A what? A door, or an opening, right? Now stuff can come in and out. Now, because you have this protein that's separating the membrane, you have an opening that allows you in and out. Now, this is a three-dimensional structure. I do it very simply here, but it looks more like this. If you look at a side view, we call it a channel protein, by the way, where the protein has this hydrophobic, obviously the inside of this has to be hydrophobic, has to be hydrophilic, must like water, because it's allowing water, the solvent water to flow through. And it has to have a hydrophobic component that resists water so that it can interact with the lipid tails. And what you get is a channel. Something allows things in it to flow in and out of certain sizes and shapes. If this is side view, if we took a top view, remember a cell would look, it looks like a big ball. Some of you actually drew it like a big ball, right? And if you took a close look at it, what you would see is these heads all around this protein that's an opening in the membrane. So this would be a top view, and this would be a side view. And I'm going to show you some pictures in a minute of cells online. I want to be able to show you the movie on here, but unfortunately I can't because better not say it, but we can't online with this internet, with the particular access we have. Using the Wi-Fi, we can't show the video, but I can show it to you on the desktop in a second. So what we can look at is we can look at some, hold on a second. We also call this when we look at the fluid mosaic model, I'm switching over now to the computer. Those of you that are watching the video, please go ahead and do a Google search for fluid mosaic model and take a look at some of the videos there. We can take a look at what they look like. And you can see what they drawn, right? Isn't that what I drew? The glycerol molecule, the phosphate with the two fatty acid chains, right? See, but this is the drawing I made. I wanted you to see the three-dimensional model. So here we go. Watch this thing. So now, do you see how it looks like a big ball? Do you see how this, and I'll post this on Schoology for those of you that are just watching this class today. You see how this is a big ball? You get closer, all of a sudden, what do you see? Oh, nothing. You see nothing because I haven't switched it over to the computer. Nobody's going to say anything, right? Only one person says nothing. I'm just standing here like an idiot and going, what do you see?
Without you, it was delayed when you were writing or something like that.
Oh, no. So it looks like a big ball, yes? Could someone say something so I know you see it? Okay. All right, so as you zoom in, what do you start to see?
What do we expect or
What do you see? What are you looking at?
I don't know, something yellow?
What are you starting to see now?
Like
Little
circles.
circles. What do you think those circles are?
The cell?
No, this whole thing is a cell. This is one cell, and we're zooming into one cell. The barrier, it is the barrier, but what do those little circles represent? What are you seeing?
The phospholipid?
The phospholipid, what part of the phospholipid?
The bilayer?
It is part of the bilayer, you're correct. No?
I don't know.
Oh, you're not thinking, that's why. You're zooming in. It is the phosphate head. You're looking at the phosphate head. Look at, let me go back to the sketch. Guys, you have to put this together. Me drawing it, and you being able to draw it is fine, but that's not what you have to be able to do. You have to actually imagine this. So you're looking at the top view, you're seeing all the phosphate heads. And there's going to be proteins in the center. There are openings. When you're looking at the side view, you cut the cell open, you see the or the lipid tails, fatty acid tails, and the phosphate heads in the side of the protein. But when you're looking at the top, the way you see in the movie, you're just zooming in and you're seeing the tops of these phosphates. Is that clear? The tops of these phospholipids. So let me switch over again and go ahead and push play. See, it's not just about being able to draw it, it's about putting it together into something that's meaningful. Now you see the heads, you see how they move? The cell moves, doesn't it? It flows. That's why we call it fluid. Do you have an iPhone? Yeah.
Yeah.
Did you update the iOS?
I don't know.
You need to update it. And then you need to make something called a focus for class that doesn't allow any notifications during class time. And when you walk into a classroom, you turn on focus. And when you leave, that's what should happen. My phone is silent. I don't even need a focus because I don't get any messages on my phone. You all should have focus on. Should be on class. You should create one for class that says no notifications. When you walk out, turn it off, and you'll get all your notifications. Then you can do whatever you want in the hallway. Does that make sense? So you see that the fluid mosaic model it shows you that this membrane is not like the wall of this room. Those windows are not going to move, right? Those windows are always going to be there. That door is always going to be there. It can't. If it moves, we're all in trouble because this whole building's collapsing. But a cell doesn't work that way. The doors and windows change all the time. That's really powerful because what that means is that every cell can make different doors on different days for different things. Oh, I need sugar? Let me put some doors in for sugar. I have too much sugar? Let me take those doors out. I need insulin? Let me put out a receptor for insulin. A little flag that says, hey, I need insulin. Oh, I need water? Let me put some doors in for water. So your cell changes these doors all the time. And it also is always moving. So watch. So now look, you see it? What are these? What are you looking at now? The bilayer. Notice there's lipids here. Here, these are phospholipids, and these are the phosphate heads. These are the lipid tails. They use yellow for phosphate, and they use green for the fatty acids. Now, what do they put? They're taking out we can actually do this. You can actually do this and see it in an electron microscope. But what you see here in this computer-generated image is like a pair of tweezers, really tiny little tweezers coming in and pushing. And if you guys go to CNIP this summer with us, if 4 of you will be invited, we do this under a microscope. You actually inject a cell with a little needle. So this is like a little needle going in and breaking open the cell. So you push it down and it bounces back up. So you can knock them around. But when you knock them around, they go back to the way they were. Why? They don't need any help because the hydrophilic will end up with the hydrophilic and the hydrophobic will end up with the hydrophobic. It's chemistry. It's not it's just the nature of these molecules. Now you see these things sticking out. These are carbohydrates. These are carbohydrates that stick out. This is very important. I'm going to talk to you about it in a minute. Then cholesterol gets put in the membrane as well. Cholesterol keeps them all separated. Believe it or not, cholesterol is very important because otherwise your cells would freeze, become brittle in the cold. Cholesterol separates them and keeps them fluid, like adding grease to metal ball bearings. It keeps things flowing smoothly. So outside you have these glycolipids and glycoproteins. Now look at this. Glyco meaning sugar proteins and sugar lipids. What are these things? These green things. They stick all the way through the membrane. So these are the doors. What do you think these green things are? Did you see them? I hope you review this material. They're proteins. They're the proteins. The channel proteins. So this stuff can get in and out. Watch. Ooh, there goes something. How'd that get out? How'd that get in? These are the doors. That gets in and out. The other stuff got bounced away. So now you're looking at a view from the outside. So you see you have the barrier, the separator, right, the membrane, the phospholipids, but then in those separators you have doors. You see how they look like from the outside? You see it's an opening. Not everything goes to the same door. Everything has its own special door. And those glycoproteins and glycolipids are very important as well. Glyco meaning sugars, so they're proteins and lipids. They have sugars that stick out. They're all different. Now have you heard of blood type? That's what blood type, that's how you define, that's how your cells can tell which cells belong to you and which are not you. Depends on which sugar they see on the outside of your cells. Each of you have a different pattern on the outside of all your cells that say, this is me. If I try to give you my liver, your body would say, this is not us, and they'd attack and kill it. We call that rejection. That's why transplant victims have to have their immune system adjusted so that they don't reject it. When they say reject, that means your body can tell that this does not belong to you. If it does not belong in your body does not want it in there. Does that make all that make sense to you? Because you're going to get a quiz next week on it, and tomorrow on this. And I don't want, tomorrow when you walk in, for bell work, by the way, so make sure you're here on time. For bell work, the quiz is going to be a 10-minute quiz on this material. You will have to draw, you will have to label, and you will have a multiple choice and matching section as well.
Are you uploading the video?
Yes, I will. Are we clear? It'd be better if you took notes down and asked questions now, rather than wait until you get home, watch the video and be confused and then come in and not do well. Yes, question?
Are we allowed to use our notes?
No, you will not be allowed to use your notes on the quiz. I'm letting you use your notes on the test, didn't I? Aren't I? But that's only because I was absent last week. And I wanted to encourage you to use your to take notes. It's the only reason. I don't believe you should use your notes on the day of the exam. I want to know what's in your head, not what you wrote down. If you don't understand, and plus, can I just give you an example? Some kids on a test that I gave for one of the classes, it was an important test, it was a big unit test, same unit test you had, except it was multiple choice and they actually took it on a computer. They cheated. They cheated on the test. I wasn't here, They pulled out their phones, they were looking stuff up. So they had the internet. But they ran out of time. Why? Yeah, it takes time to search the answers. It was a time test. They only had 80 minutes. And you don't have time to search that. They only finished half the test. Oh, they cheated. I know they cheated because the answers they found were the answers that were in the answer key, word for word. They didn't have a time to read. Why didn't they rearrange the words? I mean, that's simple, right? It's not simple to do that when you're in the middle of a test. You don't have time. The teacher, the substitute's right there. You don't want to get caught. You got the answer in one hand. You're going to write what you see. Correct? You're going to get caught cheating. That's if you have the answer, and you don't have time. So I could let you use your notes if you wanted to, and I'll put a time limit on, give you 50 questions, and still half of you would fail because you don't understand what you wrote. You have to understand the material in order to get the right answer. If you don't understand, you're not asking questions. You don't even, I asked you guys, I just gave you notes, and I pointed to the proteins. I asked you what they were. Nobody can answer. And you have your notes in front of you, and I just drew it on the board. So you have to process this information in order for it to make sense, for it to work on a test. All right.